Hi, my name is Jamie Foster. I'm a professor at the University of Florida, and my research lab is located near the Kennedy Space Center. And I study how animals and microbes communicate in the space environment. So I thought I would tell you a little bit about some of the research of why we do what we do and what we've been doing to understand how microbes and animals communicate in the spaceflight environment. So I'm going to tell you a little bit about the research we've done on the Umami mission. I'll tell you a little bit more about that. And we are working with a little animal bobtail squid, and I'll tell you a little bit about why that squid makes a perfect model for doing space biology experiments. But I can't thank my collaborator here, Eric Koch. He's really been my partner in all the research that I'm going to be telling you about today. So why should we care about the microbiology in space, in the space environment? Well, as you can see from these pictures here of these spacecraft, there are a lot of surfaces. There's a lot of opportunities for microbes to interact both with the people here working in the space environment and also the living on all these different surfaces in spacecraft. So living and working in space will require a really comprehensive understanding of the effects that the stress of being in space, whether it's the microgravity, the radiation environment, or just the mental challenges of being in a confined space for several months at a time, how does that affect the microbes that you're interfacing with on the spacecraft or the microbes that are interacting with your own body? So the questions that we're trying to understand as space biologists is what microbes are people being exposed to in this indoor setting of being in a spacecraft? And what factors about being in space, again, the microgravity, the radiation, and other factors that might be happening, what's controlling the abundance, the diversity, and the persistence of some of these microbes in that space environment, in that spacecraft? And what effects, if these microbes are getting stressed from being in space, are they having on human health? Are they potentially degrading, you know, the spacecraft around you, or are what's a normal beneficial microbe going pathogenic in the space environment? So we have a lot of questions that space biologists, space microbiologists are trying to understand about how microbes function and work in the space environment. But my research lab is kind of a little bit more focused. It's really about trying to understand how that space environment is affecting beneficial interactions between specifically microbes and animal tissues. And so here comes that squid that I talked about. So my research has been using model systems. A lot of people study the effects of astronauts and changes, but to really understand at all stages of development, humans are tough to work on in the space environment as model systems. So by using animals like a bobtail squid, we can start to really ask questions at all stages of the initiation, the maintenance, and the persistence of these kind of symbiotic interactions and how they're affected by space. So this is the bobtail squid. This is an adult, and this is what a juvenile animal looks like in the second panel here. And they have a special organ called the light organ. This is kind of there's an arrow pointing to that light organ in the juvenile animal here. And what happens is this is the site of this of where the bacteria live inside the squid. The squid has a special relationship with a bacterium called Vibrio fischeri, which is bioluminescent. And the squid cultivates or recruits bacteria from the environment to colonize this light organ, which is in the lower left-hand picture here, the scanning electron micrograph. You can see these fields of epithelial cells that are constantly beating and entraining bacteria from the environment into the vicinity of pores on the surface of that light organ. So once the bacteria colonize that light organ, they induce a whole host of developmental events. So it allows us, and things happen very fast. So the advantages of this system is that there's just one host and one bacterium, unlike our bodies that have thousands of different species interacting with it at any given time. In this light organ, there's just a binary symbiosis, making it a lot easier for us to understand how these two partners are talking to each other. And the bacteria, as they interface with these ciliated epithelial cells that you see here in these fields of in the scanning electron micrograph, are very similar to mammalian cells. And then as they enter that light organ and colonize, they start to interact with mucosal epithelium. So this type of symbiosis of a bacterium interfacing with epithelial cells is the most common symbiosis in the animal kingdom. So again, another advantage of using this symbiosis. And we can add the partners at any point in the symbiosis or take them apart. We can cure the animals with antibiotics. So all of these factors and the rapid development, the small size of these animals really make this system wonderful for doing space biology applications. So because sending things to space is very expensive, we use what's called simulated microgravity. And in the upper left-hand corner, you see something labeled LSMMG. That's low shear modeled microgravity. When a squid or a bacterium or any other organism is in this little bioreactor, um, it's rotating and the gravity is pushing down on that particular molecule, but the fluids inside is pushing up. So it counteracts the force of gravity, and it's like the squid is in free fall. It just kind of hangs out in the middle of that bioreactor, and we can kind of simulate that microgravity environment that an organism might experience. It's not perfect, but it allows us to hone our questions and ideas down. And when we did this with the squid, we found several phenotypes that were being changed under these simulated microgravity conditions. One of the first things that happens when a microbe colonizes the light organ is it starts to activate the innate immune system of the animal. And so in the squid, their version of macrophages are called hemocytes. So the first thing that happens is within hours, within about 2 hours, you start to get, as you can see here at this red arrow on this blue diagram here, you within 2 hours, these hemocytes are responding to the presence of the bacteria and bringing and infiltrating those epithelial appendages of the light organ structure. And then once the bacteria have actually colonized, they actually induce the death of those structures. Once you've recruited your bacteria, you don't need those structures anymore, and the bacteria trigger an apoptotic cell death of those structures, and then over time you regress and you're kind of left with this little bowling ball kind of looking light organ. So the hemocyte trafficking, the apoptosis, the regression are all developmental events triggered by the normal presence of the bacteria. But what we found was some of these normal developmental events change when they're exposed to simulated microgravity. For example, apoptosis happens way faster, like about 6 to 10 hours faster in the stress of microgravity than under normal, what we call unit gravity conditions. And the immune system response is delayed by several hours. So we have this kind of dysregulation of the immune system and the normal bacteria development under simulated microgravity. So we took these hypotheses of that the microgravity is changing the physiology of the relationship between the microbes and the animals, and we decided to, and we did an experiment in actual spaceflight to confirm these ideas. So that's where the umami experiment comes in. It's called Understanding the Effects of Microgravity on Animal Microbe Interactions. This was a NASA-funded project that flew in 2021. And here are just some photos of a little baby squid in our mission patch, and actually a real space squid that came and went to the International Space Station. But what I thought I would do is, for those of you who maybe have ever thought about maybe doing a science experiment in space, I'll do a little behind the scenes of how one gets a space experiment going, and then we'll go into the results of what we found in the spaceflight experiment. So, first of all, if you ever want to do a spaceflight experiment, you got to have some patience because it took about 5 years in order from the start of the concept to actually the launch of the spacecraft. So, mentally prepare yourself for delays and flexibility in your life. So we got the grant, we got the funded project in 2018, and it took a little while actually for our implementation partner, and I'll tell you what that is in just a minute. So it took a while for the documentation. We had to do risk assessments. We had to show NASA that we had the right hardware. We could do the experiment. So it takes a long time to verify that your science. Number two is called a science verification test. And I'll show you a little bit about that in a second. And then you test you do your experiment in the actual hardware before you launch into space. And at all of these steps, you got to prove to NASA that your experiment is going to work before they ever put you on a rocket. And then at the very end in 2021, here we are 3 years later, we actually do our final presentation to show that we're successful, we can do our science, and we are ready for go, meaning we are going to launch. And we did in June of 2021, and it was a short experiment. And I'll tell you a little bit about the timeline. And we got our samples back within a month of them going up to the International Space Station. So uh, and here's where we start. So we use these little mock-ups. We do what's called science verification testing. And before you build a fancy piece of hardware, we're literally using cell culture bags and putting the animals in the cell culture bag. We call those an aquarium bag. And we have motors kind of connected to the aquarium bag, and we can pump in either seawater or we can pump in bacteria and allow them to incubate, allow the microbes to initiate the colonization of the host squid and let it go for a period of time. And then we can stop the experiment and add in what we call RNA later. It's that basically is a chaotropic agent. It kind of stops all transcription and replication of the DNA. So it kind of serves as kind of like a snapshot of what that animal is experiencing at that moment in time. So it doesn't look like much, but this is how you kind of start your spaceflight experiment. And we worked with a company called TechShot. TechShot is the engineers. They can help you. You have a great science concept, and you can have what's called an implementation partner. And this is an engineering company that has a whole bunch of different flight tested, ready to go hardware that you can kind of put into put your experiment into, and then fly on to the International Space Station. So here's what it looks like when it's all polished up, where we had a rough mock-up, and here's the actual spaceflight hardware. So the squid, we use a highly technical term called bag-o squid, or an aquarium bag of squid. And the animals here, you can see we could fit about 8 of them into this cell culture bag, and we put them into boxes here. This is the company called TechShot in this blue box, and that's where all the motors are. So it's about the size of a lunchbox. So we're doing this completely automated, and we plug it into this larger box that is all automated from the ground, and this computer turns the experiment on at the right time and turns it off. And so an astronaut never has to touch anything, which is great because the astronauts are really busy. So we had these little loops, so the inoculation bag, the aquarium bag, and the RNA later bag, we had many replicates of that, and that's all confined within this, this flight hardware. And let's see, and so what we wanted to do first, it didn't advance. There we go. So what we wanted to do first, we took our bags of squid, and before we ever again launched it in a rocket, we had to prove that the hardware wouldn't impede the natural colonization, that they didn't get sick by being in the plastic bag, or they made sure they were getting enough oxygen. And so we have to, again, do what's called equipment verification testing when you do a space flight or an EVT, and prove that the animals will stay alive or your microbes will stay alive. And in this case, here we monitored the luminescence of the bacteria. I said Vibrio fischeri is bioluminescent. So when it colonizes the squid and it gets into enough high cell density, it turns on its light production, its luciferase genes, and it starts producing light. And in the absence of, so we had symbiotic animals that are showing these relative light units, this bioluminescence. And in our controls, where it was just filtered seawater, or what we call aposymbiotic animals, there was no light production. And then we took a few of these ones that were colonized, and we plated them on petri dishes, just to make sure that that light production correlated with colony forming units, and we actually showed that there were actual bacteria in there. And we also took those light organs and we stained them, and we looked for some of those developmental markers we were talking about, like hemocyte trafficking, apoptosis, and regression. And sure enough, they occurred naturally. So we were pretty confident that the hardware we built was not going to impede the natural colonization. And so we were able to move forward with our spaceflight experiment. And here is astronaut Megan MacArthur, who helped us with our experiment, putting this hardware into the International Space Station, where it could incubate and it could maintain temperature. And again, this is just another picture of one of those, we call them fluid processing cassettes, that housed all the animals, the bacteria, and the RNA later. So, and here's just an actual outline of what's actually happening on the International Space Station. So we have those bago squid, and then they get launched into space on the rocket. And then as soon as they hit microgravity, the computer tells it to start pumping in Vibrio fischeri and making those animals symbiotic. And then in a second cassette, a very separate cassette, we have aposymbiotic animals, where they're just getting filtered seawater. They're not getting any microbes. And we let them progress for 0 hours, 2, 6, and 12. And so the aposymbiotic animals are a good control to help us understand what developmental effects the bacteria are triggering in the host animal. And again, here's our bag of squid. So we had a lot of, what's great about this experiment is, and because of the small size of the squid, we're able to do lots of biological and technical replicates just in this small lunchbox size container here. So we had a total of 250 squid that we were working with in this experiment. 128 baby squid went to space, and 128 stayed on the ground. And then half were made symbiotic and half were aposymbiotic. So we had about eight replicates per treatment and 2 treatments per time point. So we had a lot of reproducibility. And then we took 4 of those animals when they came back from space, and we went ahead and did RNA-seq on them. And then we took a small cohort and we did metabolomics and lipidomics on them, so we could really capture what are the transcripts that are happening, what lipids are being made, and what metabolites are being produced in the space environment under the microgravity. So there's a lot of work that goes into building a space flight, and unfortunately you can't do every control just because you're limited by size, you're limited by astronaut time. So if you ever do want to do a spaceflight experiment, which I encourage you to do, you want to try to keep it as streamlined and simple as possible. So what happened in the spaceflight experiment? What are some of the results? So the first thing that we looked at was the transcriptomics. We took the animals that came back from space. Everything worked really well. The hardware worked well. The animals were behaved properly, and we got really great RNA out of the extracted animals that came back from space. And so the first thing that we did was we did RNA-seq analysis. where we generated millions and millions of reads. And here's our pipeline for how we mapped them onto the genome of the squid to really and quantify what genes are being differentially expressed. And then we began to annotate them. So happy to offline discuss potential pipelines if you're interested in RNA-seq analysis. But the biggest thing that we found was the biggest driver of differences in the transcriptome was the space flight. So we can see here that the ground animals that never experienced space flight kind of clustered together, and the flight also clustered together. And about 14% of that variation that we saw in the transcriptomes, we could attribute to the fact that these animals went to space flight. But what were we seeing? So we decided to make this a little more simple than looking at thousands and thousands of genes. Is we started to look at clusters of genes called gene sets. Instead of looking at individual genes, we're looking at categories of genes and seeing how these larger clusters of genes were changing throughout the time and in the spaceflight. And what we found was there is a lot of transcriptional activity when the animals, right after they're hatched and they're born, and that's very normal. We see that here on the ground. And we see a whole diverse array of transcriptional changes that are happening right away after the animal is hatching. But it's pretty similar. We did some comparisons, and the pathways that are shared in the ground and the flight are comparable. It's really after when the experiment starts to get to 2 hours, 6 hours, 12 hours, that's where we start to see divergence between the ground animals and the spaceflight animals. So right after launch, roughly they're very similar, which is good because the launching part into space didn't change the animals too much. But where we start to see the other thing we wanted to check right away is we wanted to make sure the animals that went to space were actually colonized by the microbes. So we looked at genes that are typically associated, that turn on in the host when the bacteria get inside. These are things like peptidoglycan receptor proteins, galactins, lipopolysaccharide binding proteins. These were all being turned on in the symbiotic animals compared to the aposymbiotic animals. And they're kind of evenly distributed in the ground and in the space flight. And that told us that, in fact, the animals were getting colonized in the space environment. And that's really important. That tells us that if you're going to go to space, and if you're going to bring and start to live and work in the space environment, at least your microbe can find the right place to go and colonize that epithelial tissue. So that might have important implications down the road as humans spend more long duration missions and might need to colonize new animals or even plants. So that was the first thing that we did, is we just validated that the animals were actually colonized in the space environment. But then we actually started looking at the differences that we were seeing between the ground and the flight animals. And one of the first things we saw was that things were happening faster in space flight than on the ground. Normally, the bacteria colonizes, and it takes about 6 to 12 hours for the apoptosis pathways to start to kick in, and that remodeling of those ciliated epithelial appendages start to happen. So normally in the ground, it's happening at 6 hours. You can see here a whole bunch of gene sets associated with cilia, assembly, membrane structures, motility of the cells, and microtubule formation. But a lot of that is happening faster in the spaceflight environment. So we see a lot more activity happening at 2 hours in the spaceflight, where normally in the ground things happen at 6 hours. Another interesting thing is we're seeing a downregulation in the symbiotic animals in spaceflight of things like lipid transport and lipid localization, and that comes into play a little bit later when we talk about the lipidomics. So we start to see divergence of the pathways beginning at 2 hours in the space flight compared to the ground controls. But what are we seeing? What sort of pathways? And we start to see a lot of changes in oxidative stress environment at around 6 hours between the symbiotic and the apos. And in fact, we're seeing a lot of stress at 2 hours in both symbiotic and apo-symbiotic animals. So when they have the microbes and when they don't have the microbes, we see oxidative stress at 2 hours. But by 6 hours, it's almost all only in the animals that did not receive their microbes. So in the symbiotic animals, we see a downregulation of the oxidative stress response. And then by 12 hours, it's mainly just associated with lipid transport. So let's look at that in a slightly different way. So these are oxidative stress genes. And what we're seeing is these are all the individual genes. So things associated with oxidative stress like peroxidase, the superoxide dismutase, a whole host of different oxidative stress response genes in the flight and the ground. And the orange suggests that they're actually being upregulated in the aposymbiotic animals. And this is just a scatter plot kind of showing you the distribution. So again, 2 hours, if anything, it was slightly enriched in the symbiotic animals, but by 2 hours, 6 hours, you see a much bigger spike in oxidative stress in the animals that did not receive the microbes. And in ground, we didn't really see a big change in a lot of these oxidative stress response, suggesting that the spaceflight was creating an oxidatively stressful environment for the animal, and that maybe the microbes were mitigating that stress response. We also looked and we saw evidence that gene sets associated with neurogenesis were also being upregulated in animals that received the microbes. In symbiotic animals, we saw genes associated with um, nervous system innervation, functioning of of this axon development and so forth. We saw actually enriched in the symbiotic animals around 2 hours and 6 hours after spaceflight. Whereas it's a little more variable, but we didn't see a significant change in the ground experiments. And also the lipid core. I mentioned that we saw big changes in the lipid transport genes, and we saw a lot of genes associated with scramblases and phosphatases and a few others that not only are involved with lipid transport, but are also very important in the immune response as well. And those are being upregulated in this aposymbiotic animal at not so much at 6 hours, and I don't quite have a good explanation for that, but definitely at 12 hours and 2 hours, they're enriched in the aposymbiotic animals, again, compared to the ground controls. So definitely something's changing with those lipid transport genes, but a lot of these genes, like IgGF, have a lot of other functions other than just lipid transport. So they could be playing a lot of different roles and a lot of things that are unknown, that we just don't know what those transcripts are doing. That's the biggest problem, is the most significant genes are ones that we don't know what they do. So another thing, not just the transcriptome, but we saw huge changes in the small molecules that were in the host animal in response to the presence and absence of spaceflight and the presence and absence of microbes. So again, just like in the transcriptomics, both the metabolomics and the lipidomics showed that spaceflight was the major driver of differences in the small molecules. And I'll show you just a couple examples of those of a metabolome and a lipidome change that we'll talk about. In the lipidome, one of the first things I told you, there were a lot of changes in the lipid transport, specifically a downregulation in symbiotic animals. Well, what we found is a huge amount of accumulation of ceramides during the spaceflight animals in the symbiotic conditions only. That's, as you can see here, the blue is enriched in the sim. Ceramides they are used for a lot of different functions in the cell, but one of them is linked to apoptosis. So ceramides, as they accumulate, they can amplify apoptosis signaling cascades, and we think maybe that slight increase in the symbiotic animals that we see here within 2 hours after the start of that experiment, so that the accumulation of the ceramide, and remember, lipid transport genes are down, suggesting that the ceramide is accumulating but not getting moved. So the transcriptome and the lipidome is suggesting that lipids might be accumulating in the symbiotic animal, and that might be triggering this faster apoptotic cell death of those tissues in the host animal when in the presence of the microbes. And in the ground samples, we actually see um, a higher upregulation or enrichment in the aposymbiotic animals. So a very different story between flight and ground, and this accumulation of lipids like ceramide in the symbiotic animals during spaceflight. And just one little story about what's happening in the metabolome is we saw a huge change in the tryptophan metabolism in symbiotic animals during spaceflight. So tryptophan, 95% of tryptophan metabolism occurs through a pathway called the kynurenine pathway. And tryptophan is emerging as a major regulatory pathway that microbes use to regulate the microbiota gut-brain axis. And it can have a huge impact on the central nervous system of the host. So people are really looking into this particular pathway in terms of microbiome health for animals. And in the volcano plots on the top here, you can see an overall downregulation in spaceflight of the small molecules with an enrichment in actually more in the apo than in the symbiotic ones, which are in blue here compared to the ground controls. But one pathway in particular that we zoomed in on was this kynurenine, this tryptophan metabolism. What we found is that a lot of times microbes, this is just in general, microbes will compete for that tryptophan and help regulate or lower the tryptophan concentrations in a cell so that you don't get a buildup of these kynurenine pathways. And when that happens, you can kind of get a suppression of the immune system because this kynurenine pathway, which includes all of these, I won't repeat all these long names here, like formylkynurenine. All these pathway metabolites can actually act as a suppression of the immune system. So a higher amount in the spaceflight conditions, slightly higher than in the ground. There was actually a depletion in symbiotic animals under the ground conditions, but in spaceflight it was a bit higher than ground. So the symbionts might be regulating the kynurenine pathway, help lowering tryptophan levels, help lowering some of these metabolites. It may suppress the immune system, but it might act as a way to reduce this inflammation response, the increase we saw in in apoptosis in the animals that did not receive their microbes. So let's put some of these stories together and kind of tell a synopsis of kind of what was the take-home message of this umami mission. Well, first of all, there was again an increased stress response in the animals that did not receive their symbionts. There was a sustained increase in the gene expression of things like peroxidase to dismutases when the symbionts weren't present, suggesting maybe the microbes might be mitigating some of this. There was an immune activation. I didn't quite get to show you all of those, but like the kynurenine pathway, there was a lot of increase in metabolites and genes associated with stimulating the immune system and suppressing, well, basically dysregulating the immune response in the aposymbiotic animals. And an increase in lipid transfer in the aposymbiotic animals and a decrease in lipid transfer in the symbiotic animals. So on the symbiotic side, again, we didn't see this stress response, really suggesting that the microbes might be helping to mitigate or downregulate the stress response of the animal in the space environment. An increase in neurogenesis, and we have to explore this a little bit more. Why is there an increase in expression with neuronal differentiation, axiogenesis? So are the bacteria triggering a faster development of that light organ tissue in the space environment? And again, in the lipidome, we're seeing this increase in ceramides, and that might help us explain why apoptosis under the stress of microgravity is happening faster in the symbiotic animals. So we have to further look at all of these, but the big take-home trends are that one thing is that microbes could effectively colonize host epithelial tissues during spaceflight, which was very important. And that spaceflight was the major driver of differences that we observed between the spaceflight and the ground in the transcriptome, the metabolome, and the lipidome. And the stress of being in the space environment might actually serve as a cue for the bacteria to start to initiate their developmental responses, maybe faster, but that might be helping them shape the host symbiotic tissues a little bit faster, a little bit differently in the space environment. But hopefully we have trying to build a story here that microbes might be reducing the stress, they might be reducing the inflammation of the host tissues during spaceflight and helping either restore or maintain homeostasis and resilience to those microbes in the space environment. So that's my little spaceflight experiment. I'll tell you a little bit of story about that. I really need to thank NASA for funding this project and also TechShot, which is now a company called Redwire. They're great to work with if you ever want to explore the concept of doing an experiment. Eric Koch, who did a lot of the transcriptomic work, and Tim Garrett, who helped us with the metabolomics and the lipidomics, and Anna Canessa, who at the Spanish National Research Council, who also helped us in the analysis of the transcriptome work, and also Rachel Ormsby, Ryan Bull, and Dave Reed from Redwire, who helped us actually build the spaceflight hardware to send the animals in space. And so I hope that gives you a little snapshot about what goes into building a spaceflight experiment and how you might go about trying to understand questions about microbiology in the space environment. So thank you for your time, and I appreciate any questions you have by email or by chat. Thank you.